Welcome back for our final set of sessions this afternoon. We are going to roll straight through all three sessions because we do have to stick to our 4 p.m. end time. So before I hand it off to our next panel, just one final reminder. If you still have a poster up in the poster room, we do need you to have those down by 3 p.m. today. And thank you very much for your participation in that last night. So I will turn things over to Dr. Linda Brady to get us started for the afternoon. Thanks.
Thank you and welcome to the AMP Schizophrenia panel this afternoon. I'm Linda Brady. Carlos Larari and myself co-chair the steering committee for the AMP Schizophrenia project. And our first speaker to give an overview today is Dr. Joshua Gordon, who is the director of the National Institute of Mental Health.
Oops, hello. And thank you all for coming to this session on one of the newer of the AMP projects, AMP Schizophrenia. Hopefully my slides will show up. Just push the button, there you go. Okay. So for those of you who don't know, most of you probably do, schizophrenia is poses a tremendous public health burden in the United States. And one of the features of that burden is illustrated by the graph on the right that shows a burden of disability-adjusted life years due to schizophrenia for males and females across the lifespan. And of course, the challenge with schizophrenia is that it strikes the young, typically with onset in late adolescence or early adulthood, and can be disabling throughout the lifetime. And so that's one of the biggest reasons why it poses such a burden. Now, the other feature of the disease is that it is thought to arise during early neurodevelopment. But the onset of the diagnosis is typically in that late adolescence to early adulthood period. But for some years, typically, before the diagnosis, you can detect individuals at risk for psychosis through a prodromal or attenuated phase. And it's thought that this phase is important for at least two reasons. One, of course, is that if you can capture those who are likely to develop the illness later on and address the pathophysiological processes early enough, you might be able to prevent the illness or at the very least reduce morbidity and mortality from the illness. The second feature is that it is thought, again, that these phases of illness may represent different phases of pathophysiology. And so different elements of treatment may be needed at these different phases. So with that in mind, the NIMH, partnering with a number of organizations, but I would especially like to point out the National Alliance on Mental Illness because they embarked first on an attempt to meet with both NIMH and potential private partners for a public-private partnership over the course of, actually it was much longer than 12 months, to talk about what in the mental health space and what in schizophrenia space would be amenable to such a project. During the final 12 months of those conversations, FNIH really led the effort to define the goals of a potential public-private partnership and bring it to fruition. And this partnership has been guided from the beginning by the Voices of Lived Experience with NAMI and then later with the Schizophrenia and Psychosis Action Alliance and One Mind, which is a private foundation that's also led by individuals with lived experience. We have a number of other public and private partners, and they got together to try to help define the pathophysiological processes underlying this prodromal period, with the idea that those processes could be revealed through biomarkers that we could identify, and use those biomarkers to monitor and test treatments during this early phase of illness. And I'll point out that these biomarkers are different from the kinds of biomarkers you've mostly heard of through the other AMP projects. It is challenging, if not impossible, to get at the molecular events that are going inside the brain of individuals. And typically the windows we have are post-mortem windows, and that's 20, 30, or 60 years after the onset of illness and after this phase. And so we have to rely on physiological biomarkers, behavioral biomarkers, cognitive biomarkers as a window into the pathophysiology of the illness, and you'll hear some about that today. The timeline, the project began in 2020 with an observational study of a planned 2,000 individuals. And then we are about to embark this summer on a proof of principle clinical trial or two, depending upon what we can discover through this process, with grants that will begin at that point in time. Both efforts are aimed at discovering those biomarkers and then utilizing those biomarkers to detect treatment effects. I'll point out, in addition, one other thing is that it's clear that this group that is at high risk for development of psychosis is also at high risk for other adverse psychiatric outcomes. And from the get-go, this project was designed to not just predict psychosis and also develop therapies for psychosis, but also predict other adverse outcomes with the idea that the therapies that we develop aimed at the pathophysiology of this early phase may also forestall or diminish the impact of these other adverse outcomes. The observational study is led in 3 different components. One component is the DPAC, or Data Platform with Analysis and Coordination Center, and 2 different clinical networks, one mostly focused in the United States, ProNet, and one mostly focused in a range of international sites, the Prescient Network, although in reality, they're really all spread all over, and you can imagine how difficult it is to have all-hands meetings with people spread over virtually every time zone across the globe. We have, although the observational study was hampered by the onset of COVID during this period of time, you can see by the enrollment curves that we are essentially parallel to the initial target lines, but a couple years delayed. And we have a very good success at maintaining people throughout the course of what is hoped to be a 2-year study for each individual enrolled in the study. The next phase, as I mentioned, is proof of principle clinical trials. The announcement for those were already released. The review is happening even as we speak, with the planned launch of the networks in July. In parallel to this process of awarding the clinical networks, we're also engaged in a competitive process to examine which compounds we might use for these proof of principle clinical trials, and there will be more on that in the coming months. So with that, I want to thank all the many, many people involved in this partnership. The names are too small for you to read, but it's really a fantastic group led by FNIH, but with members from across the spectrum, and especially NIMH. And I'll just emphasize again that this is a project that was started by individuals with lived experience, and it continues to be led by individuals with lived experience, including on the steering committee and even in the leadership itself. So thank you very, very much for this time, and I'm going to turn it over to Justin Baker.
Okay. So in the next 10 minutes or so, I'll talk to you about how we're beginning to really develop beyond biological assays, but really into behavioral assays. And so I'll talk about, when we talk about digital assessment, that's what we're really getting into. And because we're really just midway through the project, I'll mostly be focusing on our strategies for implementation. Here are my disclosures. So as Josh mentioned, in this project, we're focusing on really distinguishing between these different clinical endpoints. See if I can get the presentation to go. And for the digital assessments, what we're doing is we're following people across the first year of the study with a host of digital assessments. So and I think it's clear in the case of schizophrenia, but also many other chronic conditions, that digital measures, these behavioral assays, have the potential to capture data in naturalistic contents while people are living their lives. Also importantly, they're very sensitive to within-person changes over time. So if you're going to develop novel technologies to intervene, these kinds of assessments can be quite sensitive. They also, as a result, be useful in early detection of clinical changes in ways that are still very person-centered. And so to zoom in a little bit more, what we'll be getting, and then in the process of getting now, is we follow people with a wearable as well as a smartphone app for the first 12 months of enrollment. And this is happening alongside data captured from the brief psychiatric rating scale, among other clinical instruments. But I want to highlight the BPRS here briefly because it is a commonly used 24-item scale, and it can be broken down into many of those other clinical domains, as Josh was mentioning, so we can look beyond just does someone convert to a full-blown psychotic disorder, but we can also look at other things like activation, apathy, mood disturbance, disorganization, somatization, things that as someone suffering from schizophrenia would be likely to have as a symptom interfering with their ability to function. So as we move forward in these kinds of data collection, we'll have that ability to follow people in the study over that full 12 months as their levels of these factors may be changing. We'll be getting a number of ecological assessments. So here's just, you don't have to go through all the details, but each day of the study, participants are asked to answer questions about their level of energy, how down or empty they're feeling. You can read off of the list here, but importantly, these are meant to map on to these different domains of function that we could map back to things like the BPRS. In addition, we have for this study, because it's global, we've translated this set of items into 8 different languages. And also importantly, we have an open-ended diary that each day participants can actually give us more granular life experiences, context that they might want to share, which allows us to analyze both what they're saying, but also how they may be saying it as we get into analyzing the audio-video qualities from changes in participants. So in addition to those surveys, we also are collecting passive sensors from the phone. So this is what we call digital phenotyping. So I'll show some examples of how we're able to use the accelerometer or the GPS location sensor on the device, as well as the screen logs to really capture moment-by-moment changes in an individual's life while they're in the study. And then finally, we're using an Activity wearable. This is an accelerometer that's worn on the wrist. It's the same device that was used in the UK Biobank study, so there's already a large sample of data and resources around use of this device. One of the ways that we've really tried to enhance engagement in the digital phenotyping for this project was to add an element of optionality. We know that some of these data can be sensitive. Sharing your location might be something that some participants don't feel comfortable with. And so at the outset of the study, we offer participants the ability to either choose to provide all data or to provide data without the GPS sensor, or to provide surveys and voice diaries alone. And we felt that even just providing this optionality helped participants feel like they were really in control of the data that they were going to be donating to the study. With those options, here are some of the numbers of what types of procedures participants did opt in for. So a good 2/3 of the participants actually opted in for all the sensors, including the location sensor, with the knowledge that these raw sensor data were actually going to be imported directly into the NIMH data archive. So that was made explicit at the time of consent, and yet 2/3 of the participants still agreed to do that. There were a really small minority, about 10% of participants, who chose to eliminate the geolocation from the sensor that was being acquired. And then a good chunk of participants didn't feel comfortable with any passive data. So those who really wanted to opt out of that completely were around 25%. A full 2/3 of patients were willing to wear the actigraphy wearable, and then a good almost 90% were willing to do the surveys and audio diaries. Here's an example of one participant, just to remind ourselves that this is about capturing an in-depth, rich view of an actual person living with this prodromal condition. And so here, each column of data is a single day of the study. So at the top, you can see those are some of the periodic assessments, such as the initial screening visit, and then so on, like the BPRSs that are getting captured every few weeks. And then below that, you can see the participants' answers on the survey, all the different questions that they have answered over their time in the study. You can see that there is missingness, so we'll get back into that in a moment, that not everyone is going to do every survey every day. But this participant's doing a pretty good job keeping up with their surveys. Below that, you can see when they were answering their audio journals. And then below that, you can see these two charts at the bottom are showing the use of the passive sensors on the phone, and then at the bottom, the wearable. And so you can see they have different patterns of missingness, but each one provides a rich snapshot of the person's activities and sleep as relate to their symptoms. So Josh already mentioned that we're more than a year into the study since the first participants were enrolled. This is just an overall graph of where people are in the study currently as of just this past week or so. But what I want to highlight is how people have been doing with the digital measures in terms of adherence. So it's often a concern that people won't be doing these for very long, and it's true that over the first 16 weeks of the study, which is a window we chose because it may have some utility in the drug development process, you can see that about 50% of patients are still answering surveys, at least one survey a week out at the 16-week time point. And so while that's much less than what they started at, which is about 85% answering at least one survey in that first week, it's still about enough information to be able to estimate relevant signals in their clinical severity. Importantly, the adherence to surveys was not dependent or has not been dependent on the severity of BPRS. So more severe patients aren't less likely to answer the surveys, at least in the data we've looked at so far. Because even if there is some drop in adherence, as long as that's not directly linked to your severity score, then at least there's some modeling potential there. Importantly, for the passive data, we see that the passive screen data, and I'll show you quickly the other sensors, we see that has a much more gradual taper. So by about 16 weeks, there's still a little bit more than 50% of people providing some passive sensor data. And it doesn't seem to be coming off. It seems to have actually reached a plateau around 12 weeks or so. Phone accelerometer, similarly, it's a much more gradual plateau, but by and large, by 16 weeks, about 45% of participants are still providing excellent data, which means that we're really able to model sleep and activity from that. And finally, GPS, still similar to other signals. We don't suddenly see that after being in the study for a while, people are turning off their GPS sensor. It seems like the passive signals, people really do leave them on for the most part once they've joined the study. I will just mention, the potential for geolocation in this sample. We think that this provides us a way of assessing in a continuous and unobtrusive fashion an individual's community and social functioning, right? So you can identify things like hospitalizations, things like changes in a relationship, things like changes in a job, all from information we can glean through open source software like I've listed below, but basically by just analyzing the raw GPS signals that are coming from the phone. And so just to give another kind of intuition of this, here are 7 examples, not from AMP schizophrenia, but showing 7 individuals living with either schizophrenia or bipolar disorder, where each one of them shows a different pattern of location diversity, evidence of whether they're having a job or going to school consistently, as well as other significant life events such as moving between home locations or getting hospitalized or having a relationship. These are things which we think really distinguish among individuals, but also importantly are able to identify changes within, significant changes within an individual's life that could impact their symptoms or impact their need for treatment. I'll just show you a couple quick examples. These are actually from AMP schizophrenia now, just to get into, these are some examples from the wearable data. So each data set that we get from a participant allows us to really, with some fidelity, analyze the wake and sleep patterns just from the patterns of movement. We do have, operational issues on the bottom left. Some of the watches don't come back over the time scale that we expect them to and so on, but we're working very hard and we've already managed to collect, over 250 samples across the network. Importantly, though, we really think that the power of the smartphone is that you don't have to remember to wear it, you don't have to remember to bring it back. So we're able to take those same kinds of data from the phone accelerometer and the phone screen to derive wake and sleep directly from the phone sensors. And that's really important because as we began to compare data collected from the watch and phone, we can show that these kinds of measures derived from the phone are sensitive to even subtle shifts in the sleep timing, such as from the weekday to a weekend, or if you're in an episode or out of episode. So these kinds of early demonstrations show us that the kind of data we're collecting really will be suitable for downstream analysis. And then just to mention, these data are being uploaded into the AMP schizophrenia data repository. So the first data release contained data up to the first month after onboarding. And again, that includes all of the digital measures I've mentioned, as well as measures from all the other data types being collected. And that was released to the research community back in November. And then we're right in the process now of getting ready to release the next set of data, which will be multi-time points. So it will allow for those interesting analyses of how these signals are changing as clinical symptoms are changing. And so just to wrap up here, we do see that both EMA and sensors are showing an important patterns of missingness that we think encourage downstream use. And then I will just, in my last couple seconds here, mention that we are performing many different mitigation strategies to reduce non-adherence. So I mentioned the optionality. We also have robust onboarding and check-in procedures, and then this real-time monitoring of participant data to be able to address non-adherence. And then finally, we are, as was mentioned in the last session, very interested in return of individualized research results to participants as a way to motivate their behavior. So with that, I will pass it on to the next presenter.
Thank you. Valentina Mantua from FDA will give us a regulatory perspective on the project.
Hello. This advanced. Okay. Thank you so much. There's been some technical glitches in this presentation, so just want to counter that. So there are many FDA speakers in this AMP conference, and I'm glad to be one of them because that kind of, I think, speaks to the fact that the AMP Schizophrenia project is strongly motivated to deliver tools to streamline drug development. There's a strong focus on drug development. Here's my disclosure, and my opinions are my own and do not represent an official FDA position. So I'm going to talk to you about a little bit of why even FDA is so involved, because there's a lot of regulatory challenges in developing drugs for individuals that are high risk for developing psychosis, and then what AMP Schizophrenia is hoping to do to address some of these challenges, what's been the FDA contribution. But ultimately, how AMP Schizophrenia and the tools developed out of the project are going to help regulatory science to build a framework to allow clinical trials in individuals that are clinical high risk for developing psychosis. And as you can see, that's similar to the slide that Dr. Gordon showed before. There is a potential window of opportunity for drug development that corresponds to the state at which individuals are past the comorbid stage and entered stage 1, the clinical high risk. This is a population that is a clinical population. It's a population that has symptoms that can be treated. However, it hasn't gotten into that stage in which after full-blown psychosis and after repeated episodes of full-blown psychosis, then there is no return to higher functioning, and there is no return to normal in between episodes until the disease becomes chronic. And where we are now with drug development is essentially stage 2, 3, and 4. I would say mostly stage 3. So most of the patients that get included into clinical trials for schizophrenia are patients that have had multiple admissions to hospital for psychosis. Now, where we want to get to is to catch that window of opportunity, but in order to do that, there's things that need to be put in place, really. So you've seen this slide before. Everything I want to say out of these slides, that's basically an overview of what the project's aims are, is that the goal of AMP Schizophrenia is to facilitate drug development. Dr. Gordon has mentioned there's a high involvement in patients and people with living experience, and then we feel that treatments have to be delivered to them. So what are the challenges really now? First of all, as I searched into the FDA database, I could tell there were very few, I'm talking really very, very few, submissions for products that are aimed for targeting the population at clinical high risk, both commercial and research as well. And why is that? Well, first of all, the diagnosis is mostly based on clinical measures. So we have criteria that are called ultra high risk criteria, but we do not have biomarkers. So it's really hard to detect these population of patients. And even, you know, those that have tried to gather together all the knowledge and try to build prediction models, including also biological measures, really found that those prediction models have limited power in prediction of psychosis. And how do we measure outcomes in these populations? Well, you know, we could measure outcomes as what's the time to develop an episode of psychosis, but ideally we would want to have a biomarker. We would want to have either a biological biomarker or even, as mentioned before, a behavioral biomarker that is reasonably likely to predict. Reasonably likely in regulatory terms is a really high bar, I have to say, but at least something that doesn't take us to having to observe these individuals for years until something happens, right? Because as of now, clinical trials are sort of secondary prevention trials, time to event analysis approaches that follow up patients until, you know, a full episode of psychosis is coming. And but we would want, with AMP schizophrenia, to turn these clinical trials into symptomatic trials that treat the clinical high-risk state and observe for a reasonable period of time until symptoms improve. In order to do that, we really need to describe this population better than we do now. It is, you know, to make a parallel with AMP-ID, maybe it is a little bit when we were at the stage when we didn't have biomarkers for Alzheimer's disease and we were doing time to dementia analysis, but now likely we can treat symptomatically cognitive impairment even very early in the disease. So this is where we're now with antipsychotics. We're at tertiary prevention level, and the goal of treatment as of now is to reduce the impact of the disease on disability and quality of life. Where we want to get to is secondary prevention, really. Diagnose and treat the disease before it becomes advanced and disability becomes severe. And to do that, if we were to run an experiment in clinical trials, hypothetically, with our current criteria and current prediction model, the population that we would include into a clinical trial would comprise at least 4 categories. Those patients that will eventually develop psychosis, those patients that would remain symptomatic, and we haven't really characterized these ones you know, in a way that we could predict where they're going in the future, and those that will eventually remit will not have any of these. And there is an important chunk of that population, however, which is dropout. So from previous experiments, if we put all the data together in a meta-analysis, we found that the risk of developing psychosis in 3 years would be about 22%. That means that if we were following up 100 patients for 3 years, about 25 of this would develop psychosis. That's risk of overtreatment. And in the Naples 2, that's an observational cohort that's been run in North America. We found that at least 52% of patients did not complete the 2-year follow-up. That, in terms of, you know, designing clinical trials, is an extremely high burden because we need to power for a higher number of patients that we actually treat. And so we hope the AMP schizophrenia could inform the design of clinical trials and improve these challenges and streamline clinical trial design. So 3 or 4 regulatory considerations as to what we could achieve. Now, first of all, the definition of the population could be improved if we had multimodal biomarker. We don't think there's going to be one. That's actually unrealistic. It's going to be multiple biomarkers from different types of measurements, behavioral, imaging, probably genetics and clinical. And that's going to let us enrich the clinical trial population to catch those patients that will eventually develop psychosis, or at least increase the proportion of these in a way that we minimize overtreatment. There are benefit-risk considerations that need to be made if we want to treat individuals that are symptomatic. However, we don't know what their destiny is going to be. We have to have a positive benefit-risk. Drugs need to be safe if we can't predict those that will remain symptomatic yet. In terms of endpoint, to improve clinical trial efficiency, we need to have intermediate endpoints, something that stands in between when we observe patients and when they develop psychosis. And this can be, as mentioned before, either biological measures, or behavioral measures, or clinical. We could find, for example, that cognitive impairment predicts later worse outcomes. So that would be an important aim for us. There are challenges related to inclusion of pediatric patients, heard this morning. There is a prospect of direct benefit that needs to be demonstrated before anybody below 18 years of age can be enrolled into clinical trials, and that's another point to consider from a regulatory perspective. And indication, for what indication? We were discussing this before. What are we doing here? Are we preventing psychosis? Are we delaying the onset of psychosis? Are we treating symptomatically? We don't really know right now, but this is all answers that we hope to have from schizophrenia. This is where schizophrenia can contribute for. Now, first of all, I'm not going over the entire slide, maybe I don't have time, but let me tell you, this is the most diverse cohort of individuals at clinical high risk that has ever been recruited. And there's been other attempts to recruit these cohorts in North America, in China, in Europe, but schizophrenia is a complex disease in which a lot of environmental factors interact with biological and genetic factors. So it is very important to diversify these cohorts to obtain generalizability of any prediction model that we're going to develop and any biomarker that we're going to develop. So what we aim to do is that we find, as I said, probably multimodal biomarkers that allow us to describe this cohort naturalistically first. What is the trajectory of symptoms on different types of symptoms, positive symptoms, negative symptoms, cognitive symptoms in a naturalistic setting? But then we're having an interventional cohort. So how do these trajectories change with treatment? These are all information we need to have in order to model experiments, to run clinical trials, to design clinical trials. The FDA has contributed in multiple ways at the steering committee level. So we have here our Deputy Director for Psychiatry, Dr. Fish, is a member of the steering committees in the public, and the operations working group, where we've defined the project aims, redefined the endpoint definition. We harmonized the terminology to current regulatory thinking. We thought we included the psychs, which is the instrument that we use to recruit patients into the cohort, and that entered our drug development tool qualification program. The data analytics strategy working group, which is chaired by Ofer Pasternak, is in front of me right now, and where we try to develop a prediction model and a validation strategy for the data analysis. The digital biomarker and EMA working group is chaired by Justin Baker. Again, we tried to help in that one as well because we don't have experience with behavioral biomarkers. And so to conclude, as I said, drug development in population at clinical high risk for psychosis is challenging. There is substantial lack of tools, including biomarkers, to characterize this population, both from a naturalistic perspective and an interventional perspective. At the current state of knowledge, clinical trials in clinical high-risk states could be potentially very long, could require a large number of patients and therefore be unfeasible. The schizophrenia project is a first step for building the tools necessary to streamline drug development. The FDA has provided and will continue providing guidance. Schizophrenia eventually will do the same for us, will provide us with the tools to write up the regulatory science and the regulatory framework around drug development for clinical high risk for psychosis. Thank you.
Thank you very much. Gan Pandina will provide an industry perspective on digital assessments, and he's from Janssen R&D.
Thank you so much, Linda. Thank you for the organizers. And I'm here to provide a bit of a context, pharmaceutical perspective. First, I'll say the opinions I'm providing are not those of Janssen or our new name, Johnson & Johnson Innovative Medicine. They're my own. So why are biomarkers and digital measures, which I include as a part of that pool, important for neuroscience research in psychosis? Hopefully, the first three panelists have convinced you that that's true. But also, I think what I'm going to try and do is to tie this back to the neuroscience behind psychosis and the neurobiology of psychosis. So there have been incredible advances through genetic research, basic neurobiologic research, and understanding psychosis in the past 2 decades. We have identified novel targets and compounds and pathways to potentially treat symptoms, modify illness course, or prevent conversion to a psychotic disorder. But there's a critical need for us to identify, first of all, those at clinical high risk for psychosis, develop tools that we can use to do that, and also to develop treatments to treat both the symptoms and prevent conversion. These are all things that people have talked about before. Clinical trials for schizophrenia rely on, right now, still on rating scales for for assessing outcome and assessing treatment outcome. Biomarkers, including the use of digital measures, hold promise to facilitate more efficient, higher throughput clinical trials, increase signal detection, and enable precision neuroscience approaches. And that's what we're really talking about as we move forward with the clinical high-risk population. You've seen this slide before, so I'm not going to belabor much about it, other than to say the goal of AMP Schizophrenia is to facilitate drug development. So these tools are being developed in that context. Traditionally, in neuropsychiatry, we use biomarkers such as cognition, neuroimaging, electrophysiology, fluid biomarkers for understanding neural structure and function. We use them in clinical trials, and we sort of extensively characterize them and their relationship and correlation to long-term clinical outcomes. These are incredibly helpful in basic research. They're somewhat helpful in clinical trials as well. However, there's a number of challenges in implementing them, particularly when we move from phase 2 to phase 3. That is, they can be expensive, potentially time-consuming, and they require expertise and equipment. This is all the price of entry for doing basic neuroscience research, but when we move into the clinical trial space, we have a whole host of other considerations, particularly when we run large phase 3 trials. These measures can be lengthy. That can increase visit length. When we have increased visit length, we're always concerned about the impact when we're comparing drug and placebo in a clinical trial. Could that impact placebo response? There are a number of other reasons why these more traditional measures have not been used in phase 2 and phase 3 research extensively. Again, they give us a window into the brain. I can't emphasize the importance of that, and a window into the basic science behind psychosis and psychotic disorders, but they've been not of optimal value in detecting clinical drug response, and that's what we'd like to enable with novel tools. And of course, these can be instituted in large phase 3 confirmatory studies. They're being implemented right now in over 40 sites around the world in 2 large protocols with thousands of participants. But it's adding an extra burden and expense, and we want to be able to use it to detect better outcomes in our trials. So what about digital biomarkers? Can they help change the paradigm for development of new psychosis treatments? So here are the measures that Dr. Baker spoke about earlier: ecological momentary assessment, daily audio recordings, geolocation, actigraphy, vocal and facial affect analysis of on-site psychiatric interviews, and also vocal auditory diaries. So these are quite simple measures to use. Hopefully we've showed you some evidence of that. They take minimal time and effort for people to participate. They're mostly passive, which also allows us to implement them in a wide range of participants. There's frequent sampling, multiple time points, independently assessed outside of the clinical trial site. The more that we can assess people's behavior in the places where they live and they work and they go to school, the better, to complement what we learn in the lab. We know that the lab can change people's clinical states. We know that even participating in a clinical trial that's observational can change a clinical outcome. So these measures can be important for assessing those types of outcomes. However, we have less well-established relationships between these types of measures and clinical states. I put in here questionable. Some might argue that that's too strong a word, but not as strong as our traditional measures' relationship to brain structure and function or disease biology. And I think one thing that we always have to be careful of when we allow something to be voluntary in a protocol, people don't necessarily always want to volunteer for things. This does add to the burden. And because of the nature of some of the challenges that we see in patients with psychosis or emerging psychosis, allowing someone to participate voluntarily might mean that the people who have the most illness might be the most likely to opt out. So What do we think from a clinical trial perspective are of potential interest? We've told you all about the chart in red and white on the right-hand side. Individual temporal frequent analysis of subtle gradual changes in the real world, spectacularly different in terms of what the digital measures have to offer. Many measures are passive. This avoids patient filtering or subjectivity of how the measures are being utilized. Missingness provides very interesting information here. Maybe it's triggering worsening of symptoms. Maybe it's triggering a lack of engagement. Both of those could be opportunities to intervene and find out more about what's going on. Analysis of subgroups versus known disease pathways. Both Valentina and Dr. Gordon presented some data about the disease, the course of illness, and how it's cyclical in nature with clinical high-risk syndrome, that prodrome state. We also think there's rhythmicity to behavior or symptoms that might present. And then we are going to correlate all of this data with known clinician and patient-reported outcomes and our brain-based experimental paradigms. Okay, I'm going to give you two examples outside of the area of schizophrenia or clinical high risk to talk about how these might be useful in the clinical trial context. At Janssen, we conducted a clinical trial of a candidate drug in autism spectrum disorder, and we used data from eye tracking paradigms that was gathered throughout that trial, social versus non-social information, to assess people's social concentration and social attention. And we utilized that to see whether or not we could enrich our clinical trial at baseline for participants that might respond better to drug. That is, could we use this to enrich our clinical outcome in our clinical trial to detect outcomes? And what I want to just call your attention to, most of the words on this slide, you don't need to know what they mean. The stars represent, if we just used enrichment based on the ADOS score. That's the diagnostic scale that we use to diagnose autism. If we just use those ADOS scores, that star, it represents the mean change in 4 different machine learning models to confirm someone's response to the drug. If we include eye tracking data, if we add eye tracking data, that is social attention data, which is a core deficit in autism spectrum disorder, core challenge for autistic people, we enriched, almost doubled the effect size between those when we include that eye tracking data. That is, we see a much bigger placebo drug difference when we enrich patients' participation, stratify them by the severity of their social attention at baseline. What does that mean? It means potentially we can include fewer patients in the trial if we believe that social attention measured by eye tracking is a biomarker for autism. Has not yet been proven, but it's intriguing. We have similar approaches that we could take with other digital biomarkers in the CHR population. Let me give you one more example, and then I'll just go to some general questions. This is an example from mood disorder, and here we used, this is observational data of patients with depression, and they were followed by, for up to over a year, and we looked at relapse, and we used actigraphy data and self-reported mood, so PHQ-9, to use N of 1 prediction models to see whether or not we could predict when someone was going to relapse in their depression, when their depression symptoms would worsen. As you may know, in regulatory context, we often conduct relapse prevention trials. We want to see if we keep people on the drug or if we take them off the drug, how frequently or how often and how soon do their symptoms return. In this model, we were able to use actigraphy data in the same way to increase the likelihood of being able to predict relapse in depression. Again, not important that you know what sample entropy is. In this context, you can see the reference at the bottom of the slide. But we increased the accuracy of being able to predict when someone would relapse with a 71% accuracy and a false alarm rate of less than 2.3 alarms per patient per year. So how would this be helpful? Well, we could use this to model how often people might be expected to relapse, use actigraphy data to enrich that. Again, you have to believe that actigraphy data has value in predicting relapse. But again, this is another way that we might use these digital measures. And why such a large observational study with a very large sample size is important in validating whether or not these candidate digital markers could become biomarkers. So I'm going to conclude by just asking some questions. So the first thing we always ask, and Valentina asked this before, what will the compound be indicated for? Clinical high risk in and of itself is a syndrome that's impairing with functional impact, but it doesn't always convert to psychosis or a psychotic disorder. In CHR syndrome, is CHR the target? That is, are we developing a treatment for that syndrome, or is conversion to psychosis the target, or both? Could you get an indication for one and then later get an indication for another in sequence? And what's the associated neurobiology that the digital measure we think correlates to? How does it relate to those more traditional biomarkers that we use like EEG or fMRI or MRI? And then can we design a killer proof of concept study? We have to have an adequate phase 2 evidence in order to proceed to phase 3 to get an approved drug. So we use what we call the Goldilocks design. Not too big, not too long, not too slow, and not too expensive, because we often only get one chance with a compound in phase 2 to make the point. And then we want to be also careful about our analytic approach. But I want to mention that, importantly, can we proceed to phase 3? The most important stage in a drug approval is getting the drug over the finish line in order for it to be able to be used. And can the compound proceed to phase 3? Is the drug effect meaningful to patients? Does it improve how they feel or function? Is the digital measure or biomarker useful and scalable for use in phase 2? What about in phase 3? Is an indication possible, and is the regulatory path relatively clear? And lastly, is pursuing the indication feasible? We want to not just end with the drug looks like it might work. We want to end with the drug works and here's who it works for, and now it's approved. So thank you very much for your attention, and I look forward to questions on the panel.
Thank you very much. Next, we have a dual presentation on lived experience from Carlos Lorari at NAMI and Brandon Staglin from One Mind.
Thank you, Linda. Good afternoon. Again, my name is Carlos. I'm co-chair for the AMP Schizophrenia and patient advocate. And today we're going to talk about best practices and insights from the patient experience. And I'm with my colleague.
Hi, I'm Brandon Staglin. I'm president of One Mind. I serve on the steering committee of the AMP Schizophrenia. I'm also a schizophrenia survivor. Carlos and I share that. And I want to take a moment before I roll into the meat of this presentation to recognize something important. I want to recognize you. When Carlos and I went through schizophrenia and the terrible symptomatology of it, it was like facing an immense darkness that is terrifying, that is potentially life-threatening, and that is all-consuming. In darkness like that, it's essential to have hope. An essential component of hope is reason for hope. Science provides that reason for hope. The work that you're doing to help people struggling with a variety of conditions, including schizophrenia, is a source of hope for me and countless others. Carlos has written a paper called He Who Has Hope Has Everything. It's been published. He'll talk about that later. But I want to thank all of you for the hope you provided to me and so many people out there struggling with diseases like schizophrenia. You deserve a round of applause. Thank you. Thank you for all that you do. So we'll talk with you about not only why involving people with lived experience in research is essential, but also how to do it effectively for best success. So this is one of my favorite quotes from a very wise man, Albert Einstein. I was a major physics junkie as I was growing up. My life goal was to build the world's first starship. But as I grow older and learn more about life, the wisdom of this saying becomes ever more apparent to me. The only source of knowledge is experience. This is comparing abstract knowledge that you learn from a book or from school or even through science with personal firsthand involvement in something going on. And that's why you can compare psychosis as part of an example that would be applicable to this. Carlos has a great metaphor that describes why psychosis is something that can be best ascertained through experience.
Sure, I'll share it briefly, but perhaps imagine for this hypothetical you have a congenitally blind person who's a neuroscientist and studies vision, and perhaps they understand the rods and cones and the function and the structure of the eye and how it gives rise to vision and color, and perhaps they even understand, the color red symbolism in art and literature. but if they've never really experienced RED, do they truly understand it? Do they truly know it? And perhaps that's similar to psychosis. And if it was good enough for Albert, it's certainly good enough for us, hopefully, to bring in some lived experience to this work. So.
Thank you. And Albert is your middle name, right?
That's right.
That's right.
It's about as far as we have in common, I'm pretty sure.
Okay, great. That's more than that. So your learning experience today will take you through 3 stages. We'll talk about how lived experience-driven advocacy organizations have contributed to what is now the AMP Schizophrenia and how it's been formed. Then we'll talk about how engagement of lived experience in the AMP has enabled it to provide greater value for its stakeholders and advance it faster and more effectively. Then we'll talk about how lived experience can help you in your research enterprise. Many of you are already involving lived experience, but best practices to do this most effectively. So into the first section. So historically, I'll take you back to 2003. In 2003, the One Mind is an organization that is founded by my family, actually my parents, Sherry and Garen Staglin. And they partnered with the NIMH that year to convene 10 research institutions to launch an important program, later funded by the NIMH, called the North American Prodrome Longitudinal Study. Many of you are familiar with this. This was a project that actually spanned 3 studies over about 17 years to identify who was at risk for psychosis before it actually fully developed, and develop tools to predict that within about a 2-year window. It had a pretty good outcome, was able to predict psychosis using the biomarkers that it studied across the patient population, about 2,000 people studied, with about 70% sensitivity and specificity. But the biomarkers that were pioneered in terms of their study in this program now form the basis for the AMP Schizophrenia and the biomarkers that it's studying. NAMI took it a step further next.
That's right. So as you've seen, One Mind was critical in laying the scientific foundation, and NAMI was also critical in advocating for an AMP. During 2014, NAMI convened at the Stanley Center for Psychiatric Research at the Broad various stakeholders to push for an AMP. There had been an interest and appetite, but it hadn't gone off the ground. Several years later, we have the successful launch of AMP Schizophrenia, and NAMI continues to convene stakeholders to advocate for better science and research.
Well done, NAMI. Well done, Carlos.
Thank you. So I'm going to give you a whirlwind tour of our engagement as lived experience participants in the AMP Schizophrenia, so please be patient with me. So even baked into the governance and leadership, there is lived experience. As one of the co-leads myself is a person with lived experience. But what I would like to highlight here is really this kind of culture of open door policy and transparency, where we're welcomed and permitted to engage in the degree we'd like to in all aspects of the research design. And Linda really has been an exemplar of both scientific expertise and compassion when it comes to kind of welcoming people with issues that can be very challenging and sensitive to talk about at times. So we've been involved in, again, all facets from research design, reviewing the protocol, assessing patient burden, providing insight into biomarker assessment, and sharing data, including sensitive digital data, as was mentioned earlier, and data sharing processes for participants. So bear with me while I read this quote, but I think this really makes it concrete. The digital assessment platform used in AMP Schizophrenia studies, MindLamp, has been built over the last 5 years with direct co-design and over 200 updates, so its design reflects the input of many stakeholders with lived experience. This involvement, coupled with actively sharing research data from the app back to the users, has helped to build trust and contributed to the app's robust uptake by more than 80% of study participants. This surprisingly strong engagement is now yielding useful data to support the AMP's aim to lay the groundwork for developing original medicines to treat people in the earliest stages of this illness. So that's a quote from John Torres, one of our colleagues in the study, and I think this is one way that AMP is truly leading the way with creating an iterative process between people with lived experience and researchers to develop better research. We've also been engaged in the website and outreach, not just with the design layout and the health literacy of the information on there, but in other ways as well. And one thing I want to highlight here is that we're seeing significant referral traffic from the website NAMI, which suggests engagement from those with lived experience and their families. And we've really been like a little marketing engine for the AMP Schizophrenia as well, with getting the word out as much as we can in peer-reviewed publications, in podcasts, in publications for the broader advocacy community, and much more. And we hope to continue to do that. Yeah.
Awesome. So thank you, Carlos. So I'll talk with you now about best practices for involving people with lived experience in your research endeavor. This content is sourced from the HBGI, or Healthy Brains Global Initiative Lived Experience Council, a council which I spearheaded the launch of in 2020, in which Carlos is a charter and current member. These are leaders in lived experience guidance throughout the world. So here's what they have to say. Why embed lived experience in your research endeavor? So it provides a variety of benefits, including improved research quality and accessibility for the population you aim to treat, better outcomes for that population, hope and empowerment for that population, and decreased stigma and enhanced trust amongst the stakeholders involved. There are three major factors you need to get right to embed lived experience successfully in your research endeavor. The first is culture. Provide a welcoming environment to encourage robust participation by all stakeholders, including those with lived experience. Provide systems that ensure that lived experience participation is reliably and fairly sustainable within your endeavor. And then provide resources so that your team can support the lived experience participation equitably and sustainably. A deep dive on these three factors is what I'm going to go into now. There are 4 pillars of culture success: embracing diversity across different dimensions of personality and demographics and lived experience, fostering inclusivity and well-being amongst those participants with lived experience, supporting their adaptability, and seeing strengths beyond their lived experience, because people who face these terrible adversities, as we heard from the patient ambassador panel today, have unique talents and skills they bring. Capitalize on those. The second success factor is systems. Make sure you choose governance structures in your organization that are co-designed by the lived experience stakeholders and are fit for the purpose. And create avenues for open communication throughout the organization, including in ensuring that leadership is accountable for the guidance that is provided by the lived experience stakeholders. And third, resources. Support skills development by the people with lived experience in your team. Provide logistical support to ensure their full involvement, and assess your organization's resources before you start to ensure that you have the resources available to ensure their success. So moving forward,
I think we just want to reiterate our gratitude to the scientific community. As much as we try to inspire you all to continue to work hard and put care into this effort, you inspire us. It's really kind of the, as Carl Sagan would say, the candle in a dark world sometimes. At least that is for me the science. We're excited about the proof of principle study, and we hope that this can really have impact for people at high risk for psychosis. Certainly, in our stories, our treatments were delayed. Took multiple times to get the right medications and things like that, and hopefully more biomarker-informed and objective approaches to care can alleviate suffering for lots of people.
Mm-hmm. Absolutely, and I have a vision that we can accomplish together. That vision is one where everyone facing serious mental illness has a splendid shot at recovery and the full, healthy, meaningful, and productive life they deserve. I believe we can accomplish this as we work together through partnerships like the AMPS. Thank you so much.
Thank you very much. I'd like to briefly thank our FNH project manager, Loni Ajabi, for her work on the project, our NIMH team that's been just working so hard, and all of our investigators that have attended the meeting. So thank you very much, and we're open for questions. Well, while we're waiting for some questions, we have one. What are essential requirements for success in utilizing biomarkers in drug development? I know that Gan and Valentina, you both touched on that, but is there anything else you'd like to share with us?
Fortunately, my psychiatry might not be the discipline in which this is being conducted in a successful way, because we have really little understanding of the biology of the disorder. But I would say, from my perspective, it's engage with the FDA as soon as possible, talk to us, just try to reason around where are you intending to use your biomarker in your program. So that's kind of the first step and the basic step, and from there on everything is downhill, but the first thing is just to envision your biomarker, your multiple biomarkers, your whether of whatever nature that is, and try to fit it into a development by talking to us and brainstorm with us. I think that's the first step.
Yeah, I would just add, the ground truth that we've relied on for many years in psychiatry development is rating scales around symptoms that are clinician-led. And the more that we can establish with this observational trial a link between the clinical outcomes that we're typically used to measuring in the core psychosis disorder, backing them into the CHR time period and looking at how they impact outcomes and conversion rate. That observational trial is really going to be a huge boon for us. And as well as the database, I don't think we've talked about that, but the database that's been built that Naples became part of, right, the data from that to really guide the selection of those outcome measures. So, and I can tell you, working this out in the clinical drug program is not a great thing. We don't like to do that when we're in phase 3, right? We want to do that well and early on. So Valentina's point about engaging early and often around how you're going to use the marker, for what purpose, enrichment, stratification, et cetera, is critical.
Thank you. Panos?
Yes, thank you so much. So, Panos Roussos from Mount Sinai. This is an impressive effort, so I would like to say a big thank you for everything you're doing. This is really outstanding. So, my question is, over the last 10 years, there has been significant efforts to identify genetic risk factors for schizophrenia, both at the common space or rare variants. So I was just curious how you plan to use this genetic information to be able to inform your whatever models you try to build and really be able to stratify individuals based on the care that they need to get.
Thank you. We are intending to collect blood for DNA analysis in this study, and we will be thinking about generating polygenic risk scores that will feed into the data analytic plan that Ofer Pasternak and his team are leading through this project.
Rima Dauk, Duke Medical Center. 10 years of experience working with Alzheimer's disease, preceded by working on schizophrenia, bringing the power of big data and metabolomics, right? The lipidome, the metabolome. The conclusion from AMP-AD is that peripheral informs about the brain, informs about brain imaging changes, informs about cognitive changes, mood changes, depression, anxiety. I think the myth, perhaps AMP-ADs, and looking across disorders gives us a chance to revisit this necessity of looking at the brain and change the paradigm to say peripheral organs, including the exposome, the diet, the gut microbiome, all of it together, plus information about the drugs we take that taught us in schizophrenia. I look back now, 20 years ago, my journey started with schizophrenia and mapping lipid changes. We looked at 1,000 lipids. That's where the journey started. In first episode psychosis, and seeing indeed we see differences, clear differences. Then we studied the imprint of drugs, 3 drugs of schizophrenia. We saw what do they imprint metabolism in the people who respond, in the people who do not respond. So just to put an urge as we think across AMP-AD and investments that are made, that we rethink, we get a bit this innovative, we remove boundaries and think about the brain being an organ that is highly interconnected with the rest of the body, and therefore there is lots of biomarkers and changes looking now across AD and schizophrenia and PD that take us back to lipid metabolic alterations and much more.
Thank you for that input, and of course, in designing this study, there had to be tough decisions made about what to include and what to leave out. The lesson of AD is a really important one in, of course, the way you've identified that we can learn something about the function of the brain during AD by looking at peripheral biomarkers, and that's been very, very powerful. The other lesson, at least that I draw, and I apologize if it differs from the lesson that you've drawn, is that that was enabled by specific pathophysiological hypotheses that told us what to look for peripherally, as opposed to hunting in a sea of different results. And I think the attempts to hunt in schizophrenia, like the one that you embarked upon, have been modestly helpful, but not yet identified those pathophysiological processes to focus on. On the other hand, the genetics that Panos mentioned earlier have been really quite clear that a major, if not the major site, is taking place in the brain. I would add to a point that you were addressing is that factoring in immune system, whether it's immune in the brain or outside of the brain, we're not 100% sure about, is something that we'd love to layer onto this understanding. But the genetics point to developmental processes in the brain, in various cells and components of the brain. And so that's got to be our, that's what informed the starting point for this study. But we are interested in trying to understand how what we find in the brain can be mapped onto easier to measure things in the periphery.
The key is just to keep the samples. We cannot do all the time, we're clear. But if we can process, collect the samples in the right way, store them, someday we can come back and revisit. Yeah.
Josh, would you like to share any closing remarks?
Well, let me just say, this was hinted at by Carlos and Brandon, that inclusion of a psychiatric illness in the AMP portfolio was an initial goal of AMP when it was first started. And we weren't yet prepared to do that when Alzheimer's was rolled out, right? And what changed was, number one, the efforts of voices of lived experience that forced us not to forget about it, and that pulled us and the advocacy community, the philanthropic community, and the pharmaceutical community into figuring out what do we know enough about to move forward with. And then the second piece was the recognition that with the genetics and with pathophysiological identification of this phasic piece of the illness, and that an unmet need in the pharmaceutical industry in this clinical high-risk state was met nicely by the evolving science. And so FNIH then enabled us all to pull this together. And so I think it's really a wonderful example of persistence. And I'm really proud to have walked into this office just at the right time to facilitate all this groundwork that had been laid by many others, and really excited to see this project through over the next several years.